the human genome was sequenced almost 25 years ago.
It's making us both feel old, right?
Yes.
I just put you in bio category too.
In my PhD at the time, working in grad school. And at the time, it was sort of like, we have this blueprint.
Right.
And the rest is going to be so easy. Right? Now, we just need to get everyone's personalized genome, and we'll have the solution to all our problems. And that was just the tip of the iceberg.
Welcome to Holy Shift, Biomedical Breakthrough Shaping Tomorrow. What is biomedical engineering exactly? It's about engineering solutions that improve human health, from new medical imaging tools to personalized cancer treatments. Holy Shift reveals the people, passion, and purpose powering the future of biomedical research. Today, we're diving into your cell's remarkable ability to bounce back from whatever nature or our guest, Dr. Melissa Kemp, throws at them. Dr. Kemp works to decode how cells process information, make decisions, and even work together like microscopic communities. Join us as we explore how understanding and engineering living systems could lead to personalized therapies. Why don't you kick us off, Melissa, and tell us about your research, kind of how you would tell someone maybe in an elevator what you do for your research.
Yeah, so I am a computational researcher, and I simulate the way that cells behave. So that can mean how cells respond to drugs. It can be how cells organize into tissue structures. And I'm also involved with trying to understand how cells can be used as therapeutics for kind of this next generation of therapeutic design. So all of that requires computational modeling, and we use engineering principles to understand and predict the way that cells behave.
Okay, and so in this instance, then, to the audience that they're wondering, what is computational modeling? Some might know, right?
Yeah.
But this is not in a lab actually working with the cells, but working with computer technology that has been programmed to act like the cells, so that then you can advance the research much faster, right, in a system like that. Is that something?
Yeah, so we do have a wet lab, and that's an important part of what we do, because we essentially are describing a lot of biochemical reactions, or we're trying to simulate on a screen, cells that may be moving around in a dish in our incubators. So that's all computer programs that are designed to, mimic that type of behavior. So there's a lot of math involved in describing some of the biochemical reactions that take place. But ultimately, yes, it's computer code. That's the tool we work with. And we're designing computer code so that we can better predict the way cells are behaving.
Okay, and so if the listeners are wondering, okay, so how do we use that in real life? So what are some examples of the outcomes of the research that you do and how it impacts the society or healthcare?
Yeah, so a good illustrative example of how we use this is, imagine you have two children with childhood leukemia. One responds to the standard course of chemotherapeutics, the other doesn't. Why is that? Well, a researcher can go in the lab and they can measure all of the oncogenes, all of those cancer-causing mutations that might lead to an immune cell turning into a leukemia cell. And they can, try to understand sort of the underpinnings of why a leukemia cell has formed. They can take those cells, they can add chemotherapeutics in a dish and say, these cells die, these cells don't. But a lot of times those markers look identical between patients. And we know that, one patient will respond and another patient won't. And so an example that we did in our lab is we took the characterization that researchers had done, all the molecular profiling, and we did some of our own profiling, and we built these simulations to try to say, all right, if you take all these interacting parts, this network of proteins and enzymes, and put that all together and try to predict the behavior, can we understand why one patient responded and one didn't? And in fact, not only could we do that, but we could also look at, well, if you dose this a little bit different, you get an entirely different behavior. And that was sort of this really cool example of how the simulations really add this layer of insight that just would not be possible if you were just trying to,
Right.
Either just do a very widespread characterization of the cells experimentally, or very tedious biochemical, looking at one at a time, the enzymes involved. So putting it all together and writing code that really is trying to describe this behavior can really let us understand, how drugs are acting, off-target effects, dosing effects, and get toward this idea of personalized medicine.
Okay, that's what I wanted to get to, was one of these outcomes of personalized medicine. Yeah. Okay, so, but before we dig into personalized medicine, for the audience that doesn't know what that is there a certain cell line that you're most interested in, or a certain type of disease you're most interested in, or does the work that you do apply across all cell lines?
So, Angela, that's a great question. We collaborate on so many different projects. I love doing team science, and so I get excited about applying these tools to a lot of different problems. So we have things going on in the lab related to Parkinson's disease. We have simulations of inflamed lung environments. We do a lot with stem cells and looking at, their behavior. I have a longstanding interest in cancer and trying to understand how cancer cells behave. And we've applied that to different cancer types as well: leukemia, head and neck cancer, lung cancer. So really, there's sort of, every time we build a model, it has to be very specialized for that cell type.
Got it. Yes.
But we use these tools in a lot of different contexts.
Okay, that's very powerful. Mm-hmm. And when we think about, one of the things I love biomedical engineering, everyone who knows that I believe it's just the best engineering ever. Okay, I'm incredibly biased, obviously. But one of the things that I often see when I've worked with other engineering disciplines is that the key about biomedical engineering is that introduction of the human body, which it adds all these additional factors that do not exist, for example, in a circuit or something like that. And so I want to talk about personalized medicine and how valuable that is, because every human being is its own unique system that has its own factors, and being able to, through some type of model, be able to pick something for that individual is huge. Yeah. So can you tell me about a little of the work that you're doing in that, and maybe some of the outcomes that you've seen thus far?
Yeah, so the human genome was sequenced almost 25 years ago.
It's making us both feel old, right?
Yes.
I just put you in bio category, too.
In my PhD at the time, working in grad school. And at the time, it was sort of like, we have this blueprint.
Right.
And the rest is going
to be
all the
so
answers. Easy, right? Now we just need to get everyone's, personalized genome, and we'll have the solution to all our problems. And that was just the tip of the iceberg, because now we really understand that you have this blueprint of instructions that is very unique to each individual. But then you have this sort of accumulation of lifetime environmental exposures, diet, lifestyle, exercise, zip code. Are you in a high air pollution environment? What's your job? Are you a flight attendant? They're receiving gamma radiation from being at a high altitude. Are you a dry cleaner working with chemicals day in and day out? So all of those life experiences contribute to our human health and disease. And there's a fabulous group of researchers here at Emory that are involved in looking at that exposome.
Okay.
So this is a sort of a word that's coined to really capture all of that. And really sort of the holy grail is to be able to say, we understand your genetic information. We can essentially create this personalized simulator to predict your path, your trajectory towards disease, and simulate what therapeutics would would veer it back, just like a flight simulator, being able to take control and understand what the best choices are for you personally. So that's the holy grail. And so, my discipline, the computational modelers out there, are developing these tools so that eventually we can reach that goal.
Okay, I think of those movies, where there's some movie out there and it talks about like, if she made this decision, this is the way her life would be. And if she made this decision.
Yeah.
And it's that way, though. I think the information we can get from this can also help us think about impacts we might want to make for the environment or for the type of work that we do or how to improve chemicals that people work with so they have less impact on their genes in a negative way. Okay, so this is what I want to know, because computational analysis sounds really complicated. It sounds very nerdy. I want to say that, right?
It is nerdy.
So at 5, was that the nerd that you wanted to be? What did you want to be when you were 5 years old?
I was obsessed with dinosaurs when I was 5.
Oh, this is amazing.
Obsessed.
Okay.
So.
And your favorite dinosaur was?
Oh, probably Triceratops.
Okay.
I love Triceratops. Or Stegosaurus. I don't know. I love them all.
Right.
But yes, I had memorized like every dinosaur. That was even before. This was like in the 70s. Like dinosaurs really weren't mainstream. Like,
this is before Jurassic Park.
Yes. So I was really into dinosaurs. So at 5, no, I was not a computer nerd. And I actually wasn't, I stumbled upon this really when I was a nuclear engineering major in college. So I chose nuclear engineering because we did not have biomedical engineering back then. And there was medical physics and radiation sciences as part of my major, which really allowed me to take all of these engineering classes, but also cell biology and biochemistry and quantitative physiology. And so I was doing these medical applications where you can't radiate someone, right? You can't do trial and error type studies with people. So they simulate everything. It's all built into a computer, program. And so you could look at ionizing radiation effects on different tissues, how it scatters through bone versus fat tissue. And so I was doing these types of computational analyses, and I just kind of fell in love with it. And then I wanted to find a PhD program where I could continue to do computational modeling. And I wound up in bioengineering and biomedical engineering.
Amazing. Okay. Yeah. So, but I love that because you started in the one area, but you still were able to tie into the human health piece that is driving the work that you do today.
Yeah.
Now, how did you find Georgia Tech and Emory Biomedical Engineering? Like, did you immediately come here at this instance, or how long have you been with us?
I've arrived in 2006.
Okay.
And that was after a postdoc at MIT. And I was very fortunate at the time that they were seeking researchers in my discipline, systems biology. I don't think I've said that word yet, but that's really how I label myself. And so I was very fortunate to be hired here and have just continued to be amazed at my colleagues and my fellow researchers on both campuses.
So can you tell us a little bit about what partners, maybe funding sources or things like that, make your research possible?
So we get some of our research funding through National Institutes of Health, National Science Foundation, and philanthropy. Various sources like the Marcus Center, which has been created at Georgia Tech, McCamish Foundation, the Flanagan Endowment. Very generous individuals that have decided to entrust their gift into cutting edge research, and I'm very fortunate to have all of those funding sources.
Well, especially when you talked about all the different areas of this type of research, and it is not. I mean, there is value in research that is very focused in one area, and there's also value in research that has applications across a broader spectrum, which is what you have. So now, one thing I'm curious about. In this instance, where do your new ideas come from? Like, do you have a process of coming up with the next important idea to test?
Oh, that's a really good question. I am really fascinated by the complexity of biology. And I think that's really what drives a lot of what I do. It is so complicated. It's just mind-blowing. When you think of the number of cells in our body or the number of proteins in each cell, it's boggling. And we are very good at collecting data on these entities now. But putting all the data together is very complicated. And so when I think about just the orchestration of all of these reactions going on in our body in order for us to blink or think or breathe, it's just mind-boggling. So I come at problems from just sort of an appreciation of like, this is a really complicated situation. Perhaps someone's approached me with a very interesting application they're working on, and I think hard about, well, what would be the right description for this particular complex network of interacting parts? Do we want to do something that's just sort of machine learning based and try to look at how things vary collectively? Do we want to try to simulate sort of the actual molecular way that these things are colliding and utilizing sugar to make energy? When we're talking about cells sort of migrating along, thinking about just developmental processes. So there's a whole field of developmental biology that's really looking at how do you go from a fertilized egg into a whole organism, and just the events that go on with that. Thinking about how does each cell know where it needs to be and what cell type it needs to be next, and all of that. So I guess, when I'm thinking about problems, maybe it's a little bit of a long-winded answer, but I really sort of kind of come at it with an appreciation for the complexity of the problem, and then asking, how can we distill this down into something that we can describe on the computer and make predictions? And how complex does it need to be for us to answer particular questions that can push the needle forward on therapeutics?
How do you validate the answers you get to those questions, right? Because I'm thinking
about
a lot. Most of our listeners probably are now starting to figure out that you can't just put something into ChatGPT and expect it's going to be correct all the time.
Exactly.
It'll come back with something you're like, where did you get that from? So how do you, in your world, in the computational piece, validate that what it says actually translates?
This is where the ability for us to do experiments in my lab really comes in handy. So, for example, we developed this enormous pan-cancer study. So we looked at data that was already out there that had been published, where clinicians had collected samples across hundreds to thousands of cancer patients and banked that information for other researchers to use. So we were able to take that and create personalized models of each person's cancer type. And by doing that, we started seeing trends. And instead of being, I'm a breast cancer and I'm just going to be studying this cancer type, we were looking at breast cancer, we're looking at glioblastoma, we're looking at lung cancer and prostate cancer. And we're starting to see these trends that emerge across the board, specifically in the way that we thought they would be responding to radiation treatment. So we said, well, we have a hypothesis of now what's happening. Let's test this. And so then we get cells of different cancer types, some that are radiation sensitive, some that aren't, and we can test whether what we predicted from all of these personalized models would apply to these very specific conditions. So that's a great example of the type of validation, the follow-up that we do after we do a big computational analysis.
Which is huge, because if you have a form of cancer, for example, and you have cells that are radiation resistant, you want to know that, because you don't want to go through radiation if it's going to provide no value, which is what was done before we figured this out, right? You would just give everyone a particular treatment. But the ability to then model how the cells will react to different treatments Therefore, then that patient doesn't have to be subjected to the side effects and the rest of what can happen for a treatment that's not going to work for them. And instead, spend that time giving them a treatment that their cells are going to be more responsive to.
Right.
Is huge. I can think of every patient who's had, especially your cancer patients, right, who've gone from one treatment to the next to the next, and the pain and suffering of that. If you can reduce that by just a little bit,
Right.
that is life and game changing.
Yeah.
Seriously. Okay, that's exciting. So this is the Holy Shift Biomedical Breakthrough Shaping Tomorrow podcast. So I would love to know, was there a moment or has there been a moment in your science where it's been like this light bulb, holy shift moment where you saw things very differently and it's had an impact on what you've done?
Yes. I would say one example of this, and I return to this, I've even, and when I've talked at scientific meetings, I've brought this example up because very early on in my career, I had this approach that I was going to take in my lab. I was going to build these models, and then I was going to use RNA interference. So it's a way of suppressing genes and the proteins that come from the genes on a one-by-one basis. And I thought, I will build this model, and then we will know what's going to happen when we do these perturbations. It's a lot like if you were an electrical engineer and you have a circuit board and you say, all right, I've got my circuit, and now I'm going to, like, you know, what happens if
I remove this or turn this off?
Yes, it's the same type of approach. And it was a complete disaster. And we got a few publications out of it, but what I study oftentimes is metabolism. So that's how cells essentially use carbon sources, like oftentimes from sugar, to generate energy. And so it turns out that there's a lot of redundancy in the way these systems work. And if you knock down one thing, it adjusts. There's this compensatory type, I call it plasticity, but it's really this ability for adaptation to occur very quickly. Nature has designed things to really try to be, bulletproof.
Yes.
And responsive, right, to environmental pressures, stress, famine, everything. So when we tried to use these types of approaches in the lab, we noticed that we would knock one thing down. It should have been a very kind of clean, precise effect. And instead, everything was changing. And I realized, oh my goodness, this is not going to work well for what we're trying to do. And I decided to embrace that a bit. I said, you know what? Rather than me trying to create these manipulations in the cells, what if we just sort of embrace the fact that there's all this variation out there and use that natural variation as a way for us to really understand these systems. And so that has led me, down to some very interesting paths, whether you're looking at variation within a tumor, for example, variation between patients, variations between different cancer types. So I've really tried to take advantage of sort of real biological noise, if you will, in what I do, instead of trying to engineer it.
I love that. You know what it makes me think of is the example of, if you go into a room and you turn off the light, for a second, it's completely dark. But then your other senses start to pick up. And have you ever been to one of those restaurants that you eat blindfolded?
No.
Okay, I haven't. But that piece of, like, the way that the rest of our senses interact, it reminds me a lot of how in the world and the work that you do, everything else amps up in order for the survival of the system. And understanding those, because then it can have implications, right? If you treat one thing and you have all these other side effects, you need to know what they are.
Right.
Some of those might be good and some of those might not be so great. That is very cool. Okay, so can you I want to learn a little bit more about you, because I know you have a very fun hobby. So when you're not doing all this very nerdy computational stuff, what do you do outside of the lab?
One of my big hobbies is I do wheel-thrown pottery. And this is a hobby I picked up in graduate school, and it's something I've carried with me every city I've moved to, I've found a studio. And there were a few years I took off when my kids were young, and then I realized how much I missed it. So I'm not particularly good, but I love doing it. It's very tactile. I can just zone out from whatever complicated problem I'm working on at work, and just really just have fun with it. So none of these are mine, by the way.
We did miss the boat on bringing
I
one.
should have brought my own prop.
Yeah.
But I make little mugs and pitchers and flower pots and.
Well, and the beauty of pottery is that, so I've done pottery, but I've also done blacksmithing and wood turning, and they're 3 different mediums. But they all, you can think of them as like cells of a body all working together. In pottery, you add too much water, or you don't do it fast enough, or you do it too fast, or you don't have it centered. All of these types of things are very similar to the type of research that you do as well, just in a way more creative and another beautiful output. So I want to learn a little bit more about your lab. So when you're looking for, let's just say there's a grad student here that is listening to the podcast, and they definitely want to be in computational. What do you look for in team members and grad students for people to join your lab?
I look for intellectual curiosity. Number one thing. I think what we do is so open-ended, and it's oftentimes, how do we know when we've got a model that's good enough? How can we start using this in creative ways to ask brand new questions and suggest hypotheses to our experimental collaborators? And so I really I'm always looking for people that are intellectually curious, because Python coding or cell culture, those are things that can be taught. But you really want to find those people that are curious enough that they're going to be coming up with innovative ideas. They're going to be looking and understanding what's out there, what resources, even what data these days that is available for the work that we do.
You know what I think is so important about curiosity, especially in this field, is that those who are intellectually, which the intellectual part as well is very important, right? Because then when you get a result out of the computer, let's just say, for example, you want someone who's going to look at it and not just take it at face value, but say, I wonder why that happened that way. Or I wonder, does that make sense? That type of piece, to peel the onion and get a little deeper. And I think that's when some major achievements can occur.
Yes, and I think the danger of computational modeling is you can build something that, like you were asking earlier, until it's validated, that could be just total artifact, right? It's very easy to construct something that's not grounded in reality. So you always need to be questioning that. Kind of, are we making valid assumptions? Are we describing enough? Are we oversimplifying it? So all of that requires like a constant engagement with the topic.
Amazing. Okay, so if the listeners remember nothing else from our episode today, what do you really want them to take away and think about your science, the work that you do, the value of it, the impact it makes on the world?
I think that ultimately personalized medicine is something we will start seeing more and more of in our lifetimes. And it's a very exciting space to be in. And computational work is really critical to making that happen. And so, just in terms of the importance of what we do, oftentimes there's a big focus on what's happening hands-on in the lab with the experiments or the various tools that are available, whether it's a doctor or an academic researcher working in that space. But there's this back and forth that has to happen with the people that can integrate that information together and really kind of pull it together in order for us to be able to deliver on that promise. And so, ultimately that requires interdisciplinary research. You can't just be a computer scientist and do this, and you just can't be a biologist and do this, or an engineer. You really have to draw from all these different disciplines together and borrow from other fields, learn how to kind of flip-flop back and forth. Sometimes I'm engaging with mathematicians and I have to put on a different hat than when I'm engaging with a pathologist. And so, really being able to be sort of in at that intersection of all these different scientific areas.
Amazing.
Yes.
Well, thank you, Melissa, for joining us today. I am very passionate about personalized medicine. I think the closer we can get to finding the cures for each individual to reduce the side effects, to reduce the impact of anything that won't work, to speed up the cure so they can live a longer, healthier life, the better. So thank you for all the work that you do. And I want to thank all of the individuals that she mentioned that have provided funding for Dr. Kemp's lab. If you want to learn more about Dr. Kemp, please see the links to her lab and the other information in our show notes. That's it for this episode of Holy Shift, Biomedical Breakthroughs Shaping Tomorrow. If today's conversation sparked your curiosity, pass it along to someone eager to explore the power of research to change lives. As a reminder, the opinions expressed in this podcast are those of the host and guest and do not represent those of any organization mentioned. Be sure to follow or subscribe wherever you get your podcasts so you never miss an episode. To learn more about the research featured on this show, visit bme.gatech.edu or check the show notes for links and resources. Thanks for listening, and remember, behind every innovation is a purpose, a person, and a powerful story.